Everybody submitted abstracts? We're good there? Okay. One thing to, I know I said no homework over spring break. Hopefully this will be something relatively easy to think about. Everybody come up with a name for your phage. Come up with a name for your phage. And it can be something cool, okay? It doesn't have to be letters and numbers. Give it a cool name. That's the only thing you have to do over break. Alright. Actually, real quick, before we get into the article,
Can I steal a mug from your office?
of course, Grace. Alright, there are our 8 samples we're sending out. So, there were a couple that didn't give me plaques when I was trying to grow them up, so I just grabbed what I could, and you may adopt one of the strains if you would like to. The bottom one on there is an E. coli strain. I'm throwing that in for myself. So, but everything looked good. We got DNA, I think, from everything. I'm taking these over to UW today for them to sequence hopefully next week, meaning when we get back from break, hopefully we'll have our samples ready to go. All right. So that's the big goal here. All right, save that. Keep that for later. All right. So here is the paper we're discussing for today. So 2 bacteriophages. I don't know that Greek symbol. Does anybody know it? Okay, we'll call it a Greek symbol. MR299-2 and NH4 can eliminate Pseudomonas aeruginosa in the murine lung and on cystic fibrosis lung airway cells. Let's start with some basics. What were the researchers trying to investigate in this particular study, or what was their reason for studying it?
That's why I was late.
Why?
I ran into 2 dogs.
Like with a car or
No,
with
I
your
saw-
feet?
I was walking and I saw 2 dogs.
Oh, so you stopped to pet them. Okay. Okay. Thanks for the clarification. All right. What's the purpose of this paper, or what are the researchers trying to investigate? Go ahead, Annie. Get us started.
We're trying to find a way to reduce the amount of this bacteria in cystic fibrosis patients because they get chronic lung infections.
Okay. So what is cystic fibrosis? Genetic condition. There's another gene for you, CFTR, which is the receptor that is mutated in cystic fibrosis individuals. What? Yep, CFTR, cystic fibrosis transporter receptor or something like that. I don't remember exactly. But often, so in cystic fibrosis patients, they have 2 mutated copies of this, so that particular gene is non-functional. What are some hallmark characteristics of cystic fibrosis? Anybody know? Andrew?
Like lots of mucus production?
Lots of mucus production and hard to clear. So usually the mucus it's much thicker than what a normal or wild type individual would produce. These days, the life expectancy for cystic fibrosis patients, I think, is in the 20s to 30s, roughly. Even 20 years ago, the life expectancy was like to teenage, and that was it. So there have been medications that have improved the outcome. And one of the common features, then, is infections by opportunistic bacteria like Pseudomonas aeruginosa. So with the thick mucus that gets produced, it makes it very challenging for individuals who have cystic fibrosis to be able to clear the mucus and to clear the bacteria from the airways. Okay, why? So that's kind of the reason to look at this in the first place. But why look at bacteriophage as opposed to antibiotics or some other therapy? Tori.
They have the ability to like break through the biofilm.
Yeah, so phage have this. One of the things they talk about is phage have the ability to break through biofilms. What kind of enzymes or mechanisms would they use for that?
The arcoiphilum that was like a polysaccharide
Yeah, so the biofilm itself is one of the terms that gets used sometimes is exopolysaccharide. Okay, what kind of enzymes do we know that break down, for example, the polysaccharide or the peptidoglycan of bacterial cell walls? Lysozyme. And so you could argue, or at least this is how some have argued, is actually the base plate of these bacteriophage contain enzymes that allow them to eat through those biofilms. Okay. So that's one benefit. What's true about bacteria that are found within biofilms? What's true about their response to antibiotics?
like antibiotics don't really get rid of them.
Yeah, usually bacteria that are found in biofilms are actually much more resistant than cells that are free-floating. Okay, so the benefit here would be use the phage as a way to basically dig into those biofilms, and then the phage can infect, replicate, kill those host cells. That's the design behind it. Okay. Other reasons why this may be important too is that for Pseudomonas aeruginosa, for example, there's been a rise in antibiotic resistance. Drugs are because of mutations that have arisen in the genome. And then Pseudomonas is also just pretty naturally resistant. One, because of biofilms. Another reason, it forms this mucoid or thick capsule around it which protects it from antibiotics. And then third, this Pseudomonas aeruginosa encodes what's known as a multi-drug resistance pump, or what's sometimes referred to as an MDR. And the one specifically is called OPRM. And OPRM is an active efflux pump, meaning as soon as antibiotics come in, ATP is used to pump those antibiotics right back out of the cell. Right? And it's been shown that if you knock out this gene or you mutate this gene, Pseudomonas loses or it becomes more sensitive to some antibiotics, not all, but some of them. All right. Here's just an image on the bottom here of, we've talked about this a little bit already, but this is actually from, I think it might be from a lung biopsy if I'm not mistaken. And you can see most of this is a pinkish color, but then there are kind of like these darker pink areas in the middle. Those represent the Pseudomonas aeruginosa, and what's around them is that mucoid layer that protects them from antibiotics and from host defenses as well. What are the characteristics of the phage that they identify in this paper? What are both the phenotypic and some of the genotypic characteristics that they identify? Go ahead, Andrew.
Or one of them most likely to be a potovirus?
Yeah, one of them is a Potoviridae. So which one fits that description better based on what you see up here, or based on these two images? What's that?
Is it the MR290?
Yeah, so what's characteristic about Potoviridae?
Short tail.
Short tails, yeah. So this is, you've got the, and they kind of point this out. So here is the tail, and I should use a different color for this today. Bear with me. There we go. Okay. So short tail, Big head. Kind of looks like a balloon almost in some ways. All right. And then NH4, what kind is this one? It was, yeah. And there's a couple of things that they point out. So one, here's that capsid head structure. Here's that tail. And they actually say inside of here is that sheath that lies underneath that contractile tail. So that they're showing this image where those tails have actually contracted, and you've got the sheath, which is what allows for that DNA to make it from the capsid head into the host cell. So one myoviridae, one podoviridae. Obviously, we can't see everything in these images. It's easy to pick out the capsid. It's fairly easy to pick out the tail, but the tail fibers are a little bit more challenging, and sometimes they just get broken off because they're relatively fragile. So that's just something to note real quickly. Size of the capsid head, relatively similar to one another. So just note the scale bar down here is a 50 nanometer scale bar, which would allow us to predict at least the genome should be relatively similar in size. Again, the capsid's not going to be much larger than what's required to fit the size or to be able to package that genome. Okay. What are characteristics of the genome that they identify? There we are. I'll go back a couple slides in a minute. But what are some characteristics of this genome? Double-stranded DNA, which is not uncommon. Let's point out a couple of the proteins in here and just kind of describe what their function would be. Hey, this one encodes DNA polymerase. So does this one. What's DNA polymerase required for? Replication. I figured we'd get us started on an easy one. Okay. How about both of these encode a terminase? We've talked about what the terminase does, but we haven't necessarily called it by name. Anybody know what the terminase is? So this is the one that actually will recognize, after you have rolling circle replication and get the concatemers, the terminase is the one that's actually making the cuts to resolve those concatemers into the individual genomes. So it's involved in also creating those terminal ends, and then also in packaging that genome into the capsid head. So this would be considered to be a late gene. Okay. And note what it's located by as well. It's located near, at least in the top one, a major head protein and a lysozyme, both which we'd expect to be late gene expression. Okay. What else is unique about the one on the top? What other features does it encode that aren't necessarily protein? Yeah, it encodes its own tRNAs. It has 3 of them, suggesting that there might be bias toward certain codons that are not as well recognized in the host cell. What do the colors in this graph mean? So there's some red, there's some orange, there's some white, there's one that is orange and white, which is located right here. What do those colors mean? What's that?
Protein level?
Not protein level. It does tell us something about the protein, though, or about at least about the open reading frame. Annie, what do they tell us?
Similarity to the phage that's like most closely related.
Yeah, so this is an annotation. I mean, this is a partial annotation that they've done, and what they've done is they've taken a look at each of these open reading frames, and they've identified, in comparison to the phage which one is the top one again? Can't remember off the top of my head. It's the podoviridae, the MR, and then the NH is here. So when comparing MR to its most closely related phage based on sequence similarity, they look at how similar is the amino acid sequence for these open reading frames. And so red means that there's very, very high similarity, 95 to 100% identity. Okay. Orange, 90 to 95%, white, less than 90%, and then the hash marked one means there's no similar gene in that most closely related strain. Maybe in others, but at least in the most closely related one, there doesn't seem to be. They refer to in the paper transcriptional blocks. What do they mean by that? And with respect to the genome, what do they mean by the term transcriptional block? And if you're having trouble, I'll circle one to maybe help you identify what they mean. So here's one transcriptional block. Here's another potential transcriptional block. Not just the open reading frames, but what's true about the orientation of those open reading frames? Yeah, and so the one here that I'm labeling as A, note that all those open reading frames are actually in the same direction, whereas this second one I'm labeling B is in the opposite direction. Those are what they're referring to as transcriptional blocks. What you'd expect in these is that there's probably genes that are expressed at roughly the same time. So all are immediate earlier, all are late genes or something like that. You also might expect that they're referring to them as blocks in part, I think, because you might argue that these have come from a different phage, or might suggest a recombination event between two phages, something like that. This, again, is not an unusual feature that we might find multiple open reading frames in the same direction, and maybe even under the control of one single promoter as well, if it's a or if it's a polycistronic mRNA that's being produced. All right. I'm going to come back to this table in a little bit, but I just wanted to reference it real quick. Let's talk about the Lux system. What is the Lux system? In the simplest form? Yeah. You know that problem that I had on the exam about luciferase? Luciferase is part of the Lux system. Okay? And this is commonly used by Pseudomonas. There are other bacteria that have systems like this as well. Vibrio fischeri is one that allow bobtail squid at night. They kind of glow a little bit, which helps them to identify food at the bottom of the sea. They have a Lux system as well. And in this particular system, what they've done is they've just taken this vector or this plasmid encoding Lux genes, C, D, A, B, and E. They've put it into Pseudomonas in this case, and that allows for them to autofluoresce. What's the benefit of this? Can detect if it's present and levels as well, because we can quantify the amount of light. We do it in terms of what we call relative light units. Grace?
It gives you like a real-time picture of like what's going on.
Absolutely true. So they do these experiments where they track from one point to the next what's going on. You don't have to disrupt the biofilms to be able to do that experiment. All you do is just put it into a little plate reader and read how much light is coming off of it. That's a really good point. It also allows you to do these experiments without, as I just said, without disrupting the biofilm. But sometimes it can be very hard to remove bacteria from a biofilm. And so this actually can serve as a way to accurately count or give you an accurate measurement of how much bacteria is there. What we're assuming in these experiments, though, is that each cell will equally express this Lux system, so will fluoresce equally well regardless of which cell it is. Go ahead, Grace.
So kind of what I got out of it and what I read is that it actually was like gram-negative bacteria in the 16S region. Is that true?
Oh, I don't know.
Oh, okay.
Good question. I have no clue. I do know that in the natural making of this system, so like in Vibrio fischeri, for example, this Lux system is based on quorum sensing. In other words, there's a critical threshold of bacteria that need to be all in one area for this process to occur. But the way that they're doing this, if I'm understanding this correctly, I think it's independent of how much bacteria is there, that this is just going to be expressed at all times. Okay, I've done that figure. All right, so here is that Lux system in practice. Here's what we'll do. I want you guys to talk with somebody else about what are they doing in this figure. So 2 minutes, describe what's being performed in this particular experiment that they show in figure 3. Go. What are they testing in this experiment?
What is RLU?
Relative light unit. So it is basically, since it's kind of like absorbance, there's no unit on amount of light. So what they are, it's kind of like I don't know how to best describe this. Basically, you have a camera over the top or something like that. It calculates how much light takes to hit, or how much light hits it in a period of time. That's your relative light unit. So what are they actually measuring there, Morgan? What are they measuring the light of?
Oh, from my understanding, the P fluorescence gives off the light, and the less light given off, the more that we just be able to destroy it.
Yes, so that light represents the bacteria in this experiment. Yeah, absolutely. And we'll look at that in later experiments. Figure 4, 5, and 6 all look at the effect of the phage in reducing that amount of light, so reducing the amount of bacteria that has adhered to a plate, for example. And this one, this is without phage. What are they looking at in this particular experiment?
Just the growth of the biofilm.
Yeah, growth of the biofilm, right? So inoculate, take a picture after 1, 5, 24 hours. 24 hours after having washed it. What's the benefit of washing here? Yeah, it gets rid of the planktonic cells, so those that are free-floating. So doing that experiment just says, hey, most of the bacteria that are in these wells are associated with a biofilm. And they've quantified that data on the right-hand side. All right, that takes us to figure 4. I'll dim the lights for this one. What is calcifluor white?
Isn't that what they use to stain cells with?
They do use it for staining, but it's not staining the cells per se. There's a short little blurb in, I can't remember if it's in the paper itself or in the results or if it's in the methods section. It's also, yeah, it's also on, no, you're right, page 4 is where that information is. So what does it say? Oh, I just highlighted this again last night.
So it's a fluorescence enhancer that binds to beta and beta and how is that right? Like it just found in biofilm matrices.
So in other words, it's binding to the biofilm, right? So here are, and the way that they've done this is they have basically scraped these biofilms off of a monolayer of cystic fibrosis cells. They've put it onto a glass slide, added the calcifluor white, and now taken images of it. This is in part a qualitative measurement. So in this one, you really have to be observant and kind of do your best to describe or explain what do you actually observe. So A and B represent two different strains of Pseudomonas without the bacteriophage added. C and D represent the same two strains but with the bacteriophage added. What do we observe?
Less of the calcifluor white when the phage is present.
Yeah, we could argue that there's less of calcifluor white. It'd be nice if we could quantify that in some way. What else? I think that's the big takeaway. I mean, there's not much more to say than that. What do the results on the right show? After 20, so monophage present after 24 hours, and we observe a general increase. What's likely contributing to that increase? Don't think too hard about this one. Phage life cycle. These are lytic phage. They're going into those biofilms. They're not only breaking down the biofilms, but this data indicates that they are capable of infecting and replicating within those host cells, within Pseudomonas aeruginosa. How do we know that these aren't lysogenic?
24-hour period.
What's that?
24-hour period.
So we know that they replicate. What might we have looked for genetically to suggest that they are or are not?
One in the genome.
Something that's analogous to C1. What's that? Yeah, the integrase problem kind of got at this as well. So integrase isn't something we naturally talked about, but we know that for a temperate phage, it has to integrate its genome into the host genome. Therefore, it has to have an integrase or some similar type of enzyme to be able to perform that function. All right, let's take a look at figure 5. Somebody willing to explain what they did in this experiment or how they did this experiment?
They had a control and experimental group.
Okay,
One,
great start.
thank you. One that used the phage as treatment on mice, and then one that did not use the phage as treatment.
You're looking at figure 6
And,
with mice.
oh, dang.
This isn't mice yet, but they still have those control grays.
Okay.
So I'm going to give you that. So, hey, no treatment with phage. Minus plus. What are they looking at here again? So go back to figure 3. What were they looking at in figure 3?
Growth of Pseudomonas biofilm.
Good. And these ones have been established. So these are ones that have been grown for 24 hours ahead of time. And they're calling this time 0. That 0 here refers to just the amount of time in contact with phage. Right? So the biofilm has been established for 24 hours. What do we observe happens over time in these two different Pseudomonas strains, here and here, as they have contact with the bacteriophage? Decrease in what?
Biofilm.
Or be more specific. What's fluorescing here?
Pseudomonas.
Okay, good. So there's less Pseudomonas. We know that for certain. And that not only do they determine this based on showing us the image, but what else have they done? They've quantified it. And that's what this image over here is, the graph on the left-hand side represents. So quantifying amount of fluorescence before and after, and it decreases by how much? 2 logs, roughly. You could say that. You could say it decreases by 100-fold. You could also say there's a 99% reduction in the amount of light. We don't know if this necessarily correlates with that much of a reduction in the Pseudomonas bacteria, but we at least know from these results that there is an effect. Okay. And now they move to mice. All right. So all this has been a lead up to show that yes, these two phage are capable of killing of destroying biofilms that have been created by Pseudomonas aeruginosa. We're now going to go into a mouse model. Oops. And take a look at what happened to these mice without or with phage being added to those mice after pre-infecting with Pseudomonas aeruginosa. Here's another powerful part of the Lux system. You can do this kind of imaging live in a model organism, whereas you can't do that without killing the mouse with other mechanisms. So they perform this experiment. Again, the blue represents some light. As it becomes green to red, that represents an increase in the amount of light that's being produced. What is happening here?
Not treated with phage, the cystic fibrosis spreads, or well, the Pseudomonas spreads. Treated
Okay.
with phage, it decreases.
I would not disagree. So we see there's an increase in the amount of fluorescence in these mice, and they actually do glow if you were to put them into a dark room, okay? In these conditions after 8 hours, and then just as you said, Grace, with the phage, there's a decrease. Looks great, right?
Yeah.
Exactly what we'd expect. What are the drawbacks of this experiment, though? And they do allude to this not only in the results but the discussion. They recognize that there's a key fault in this experimental design. I'll give you guys a minute or two to look through it. Particularly where you may want to look is
Is it the
5 and like end of 5, beginning of page 6. Go ahead, Grace.
I don't know if this is right, but that if you administer the phage 2 hours after the infection, it's not effective.
Well, they've shown that it is effective after 2 hours of the administration. But it does have something to do with timing. I'm going to go back to another picture real quick. I'm going to go back to figure 3.
Is it the glutamics?
What do you mean?
I don't know.
Go with that thought, though. So what did we say was true in this figure here? We said that over 24 hours a biofilm is formed. What's happening at 1 and 5 hours, though? So look at the 1 hours real quick. What's the fluorescence like in here? Huh? Low. Has a biofilm been formed? There's the fault in the experiment. And they do recognize that after 2 hours, it may not be enough time to have allowed for biofilms to be established. We know that these bacteriophages do have the ability to break down and establish biofilm. They show that in figure 5. But the fault in this last experiment here is that they don't give it enough time to actually be established in the lungs of those mice. So, go ahead, Annie.
Why didn't they do it after like 24 hours?
That's a good question. Wasn't a good model. It wasn't a good model to be able to do that. I think if you were at the concentrations that they're working with Pseudomonas, I'm going to bet it would have been debilitating to the mice within 24 hours. Probably would have been considered to be an unethical experiment. But there's always... Oh, damn it, Lord.
What'd you do?
Nothing. I just didn't include what I wanted to.
Yeah, that sucks.
Don't worry. We're going to figure it out. They end up doing that, so not them, but another group ends up doing this experiment. Are they breaking down those biofilms and killing the bacteria? Yes. So now what they end up doing, this is a different group of individuals, but what they ended up doing was using a different mouse line, which serves as a good model of cystic fibrosis. And they use these two different strains of bacteria. And let me set up for you what they've done in this experiment then. So in this experiment here, that doesn't really work. Yeah. All right, so in this experiment here, what they've done is they've taken these mice, they've taken 10 randomly into 2 groups. They've administered the Pseudomonas, they've allowed it to incubate inside the mice for 72 hours. That's enough time to establish a biofilm. And now what they do is after 24 and 36 hours, so 2 rounds they administer phage, and then they measure after 48 hours what's the concentration of Pseudomonas aeruginosa. This experiment is different. They actually sack the mice, they grind up the lungs, and then they measure in the lungs how much Pseudomonas aeruginosa is there. Okay? So control group has been left untreated, phage-treated group 24 and 36 hours, harvest both at 48 hours to measure how much Pseudomonas is there. They do this again, but administer at 72, administer the phage after 72 and 86 additional, or 84 additional hours. This last one is after 144 and after 156 additional hours. In other words, they're looking at what's happening after one additional day, 3 additional days, or what would that be, 5 additional days of allowing for the Pseudomonas to incubate. In general, what is the trend here?
Significant decrease in Pseudomonas.
Yeah, and probably, this is probably their limit of detection here, going down 4 logs. That's a 99.99% reduction. Does the effectiveness of the treatment change with the amount of time Pseudomonas has had to incubate?
Not according to their 3 little stars.
The 3 little stars at least indicates between this one and this one that there is a significant difference. What if we were to compare these ones to each other? Does time have an effect?
Yeah.
We could say that it does. There's 3 right here, 3 mice that seem to be unaffected. It seems like maybe the effectiveness of this therapy, and even though there's a reduction here, the effectiveness of the therapy maybe changes slightly as Pseudomonas has longer times of incubation, allowing for those biofilms to become even more hardy and protect those cells even better. But overall, the general trend is, hey, this works on established biofilms. Okay. Any questions with this data? All right, so the last question to think about, and this goes back to something I asked as part of the critical thing, or the lit review. how would this data have looked different if they had used different strains of Pseudomonas aeruginosa? So how would the data look if they had used, and the ones that they began with, again, are MR, for goodness sakes. Well, just look in your handout. I will read out the names. Because I'm done with technology. It's spring break. Yay! All right. They use MR299. They use NH573888A. And those both are ones that are considered to be sensitive to the two bacteriophages that they use. They either receive a 3+ or a 2+ score. What if they had used MR300 or MR325? How would the results of this experiment have differed if they had tried this with those phage, with those bacteria strains instead of the ones that they did use? Go ahead, Andrew.
Those two strains of P. aeruginosa are much less sensitive to the phage.
Good.
So the experiments there would probably be a lot less lysis going on, and a lot more light throughout the.
Beautiful, beautiful. They would be more resistant to the bacteriophage, right? So what this goes to show is that even within strains of the same species, we can get variability in how effective bacteriophage are. Bill and Andrew, you and Amelia and Morgan, you guys can't answer this question, so that takes out half of everybody. Why are they using two phages here? Grace?
Because, you would need two independent mutations in order for the
Beautiful.
bacteria.
Yeah, target two different receptors, and so that if the bacteria have gained resistance to one of the phage, they haven't automatically gained resistance to the other as well. So absolutely, you need two independent mutations to gain resistance to both of those phages. That's the idea of any cocktail therapy. Retroviral therapy, which we'll take a look at the end of the semester, works very much the same way. All right. Any last questions? All right, have a great spring break. I'll see you guys when you get back. Enjoy the weather, enjoy work, etc. Nope, I'll be paying.